dear friend and colleague, Adrienne. Adrienne did her undergraduate in Michigan, so she was commenting how this is peanuts compared to Michigan weather. And then went to New York City and did her Ph.D. under the supervision of Andrew Baker at Columbia University. And then he moved to Rasmus, Miami, where Liana and I did our PhD. She was, I heard her say she was squatting there. She wasn't really formally a Rasmus student. She was a Columbia University student. And so she was at both places. And then from there, she went to Oregon State University, where she did a postdoc with our friend and colleague, Becky Vega, whom you've met in one of these seminars many years ago now, actually. Wow. And now she's an assistant professor kicking butt at Rice University. So, take it away. Thank you.
Well, hold on, actually. Thank you all so much. Thank you for introducing me and inviting me, Monica. It's been wonderful already to get to talk with everyone. And I'm excited to share some of the research that my lab has been working on in terms of multi-partner interactions in threatened coral hosts and reef ecosystems. And it'd be great to talk with you more about this and hear about what you're doing later today. Okay, so coral reefs are these productive and highly biodiverse ecosystems that stretch hundreds of kilometers that you can see from space, but they are fundamentally constructed by these tiny organisms. So if you zoom in, you can see that they're made predominantly of coral colonies. And if we zoom in even further into one of these branches, you can see the fundamental clonal unit of a coral colony, which is a polyp. And inside these polyps, much of their coloration comes from the millions of microbial symbionts that they harbor, and in particular, these dinoflagellate endosymbionts in the family Symbiodiniaceae. And this relationship, this nutritional symbiosis between corals and their dinoflagellates is really well studied because essentially what happens is the dinoflagellates are photosynthesizing, they produce sugars that they release many of those sugars into their hosts. And that energetically subsidizes the coral animal to construct these gigantic reef frameworks. And in turn, the Symbiodiniaceae are receiving nutrients from coral wastes, waste products, and a sheltered place to live. But there are risks inherent to being part of an obligate symbiosis, namely that if, for example, environmental conditions become too stressful, that relationship can break apart. And so with Symbiodiniaceae and corals, we see that often in terms of a process called bleaching, which you can see here, or not. Is that the point? Nope. Notice what I'm doing. Not that. Oh, here we go. Okay, anyway, what you can see here. So in bleaching, that's a mass loss of those dinoflagellates. So here are pictures of the same reef over time. This is the reef when it's healthy, and it has all of these, it has a golden brown color because of all those symbionts. Unfortunately, during this third global bleaching event, conditions became too hot, the symbiosis broke down, and the symbionts were lost. So here there's living coral tissue, and you can see the calcium carbonate skeleton through that tissue. And so at this point, what can happen is either the coral can recover its symbiotic complement and hopefully go back to being healthy, or if that doesn't happen, then eventually this reef, these corals will die and be overgrown by something else like these dark brown fuzzy macroalgae or turf algae here. So unfortunately, because we're so many environmental changes and stressors are occurring, these sorts of microbial host interactions are having lots of breakdowns like bleaching, and that's contributing to reef decline on a global scale. But as important as this dinoflagellate coral relationship is, those dinoflagellate endosymbionts are not the only microbial symbionts present in the coral host. So maybe as members of a microbiome center, I don't know, when I say microbial symbiont, maybe you think just bacteria. For the purpose of this talk, I'm using microbial symbiont to refer to anything that you'd have to look at with a microscope. So that could include the dinoflagellates, viruses, et cetera. And all of these partners are here in the coral colony, and these other groups like the bacteria and viruses we know are also diverse. And so because of this, corals have been described as potentially the world's most diverse symbiotic ecosystem. And to me, this is amazingly compelling to think about how all of these different interactions between these symbionts can occur with the host, and also how potentially these symbionts could be interacting with each other as well. So given that interest, I really liked this new Matthews figure that was just published in a mini review in Environmental Microbiology. And it is basically just a schematic drawing of what I just showed you with the coral nigerian here, and then all of these potential different interactions with other microbial symbionts depicted with these lines. So in their review, they're showing the solid lines to indicate that most research activity has occurred between looking at bacteria coral interactions or Symbiodiniaceae coral interactions. And all of these other potential interactions haven't been nearly as well studied. And they're highlighting with this red line the connections between bacteria and Symbiodiniaceae, because there are many reasons to think that there could be important metabolic exchanges or communications happening between Symbiodiniaceae and bacteria. So they're calling researchers to place more effort in this area, which I completely agree with, but I also want to look at connections between some of these other groups. So this slide is essentially just to show you some of the areas where we currently have projects with lab members, and to give a shout out to the awesome people in my team who are making all of these things happen. So if any of these interactions catch your eye in red, I'd be excited to tell you what we have going on or talk with you about your project. So what I'm going to do today, hopefully in the correct amount of time, is show you 3 stories from projects that we've started in this multi-partner interaction vein. So the first is looking at Symbiodiniaceae community characteristics as potential indicators of host stress tolerance. The next is just showing you where we're at in trying to understand how viruses may interact with other symbionts and potentially influence the holobiont, which is the host plus all of the symbionts on and in it. And then finally, I will have just time for a teaser on this one, but thinking about where and how corals get their symbionts from this multi-partner interaction perspective. Okay, so for this first one, Lauren Hauker, who's a Ph.D. student in the lab, led this work. And the paper is going to come out in Global Change Biology next week. Okay, so here we are again with corals and their Symbiodiniaceae. When you are just looking at Symbiodiniaceae cells, they don't necessarily look very morphologically distinct, but they are incredibly genetically diverse. And here I'm just using colors to represent that genetic diversity. And anytime that you have a coral species that's capable of interacting with multiple Symbiodiniaceae strains or species or types, then that is potentially an interesting place to look in terms of community responses, in terms of acclimatizing to stressors like climate change. So for many coral species, they typically harbor 1 to 2 dominant kinds of Symbiodiniaceae, and then they may have other kinds of Symbiodiniaceae at relatively lower abundances. So to depict that, I'm going to use this little coral icon with all the dots. Okay, so here is our coral holobiont, and we know that individual holobionts can vary in their response to stress. So in this middle panel, this is going to show some type of environmental stress and a potential microbial community response. So one option when this holobiont experiences stress is that it basically is able to just keep everything under control. The microbial community doesn't change. This y-axis represents just some sort of community change metric over time. So there's no change that occurs here. The coral remains healthy and there's stability. Another option where you could see stability is a short-term resilience mechanism where essentially there is a brief shift in the microbial community, but it quickly returns to the pre-stress community. Another option where you get turnover is that the microbial community could shift And that shift could be beneficial. So it could be that the microbial community change allows the coral holobiont to acclimatize to the stress and basically just accommodate whatever environmental change has occurred. Or you could have dysbiosis, in which there's turnover, but the microbial community is becoming more dysbiotic or imbalanced. And in these types of cases, what we typically see is a decline in host health and or disease. So when we're looking at these kinds of microbial community level changes, there are different diversity metrics that we can use. One is alpha diversity, which is just looking at the species that are present in a sample and how evenly balanced they are. And another option is to look at beta diversity. So with these, we typically use these two-dimensional principal coordinate plots in which one dot represents the microbial community within a sample. And so these colors represent some group of samples. And so two dots that are very close together in space have very similar microbial communities, and dots that are far away from each other in space have very different microbial communities. So one possibility is that you could see significant differences in the community composition among groups. And another possibility is that you could see increased variability or dispersion within groups, which is also described as distance to centroid. So now we can make the colors represent stress level. And so here, a lot of microbial ecology has looked at deterministic level changes with the idea that stress is going to cause the microbial community to make some sort of signature shift to another phase, so that you would have sort of a healthy signature move to a more stressed signature to a very stressed or declining health signature. But another possibility is that a stressor could cause more stochastic changes in which you're just generating greater variability among microbial communities or more dispersion. So this idea has been called the Anna Karenina principle, based on the Tolstoy quote that all happy families are alike, but each unhappy family is unhappy in its own way. So if you transfer that to microbiomes, it's sort of like having that all healthy microbiomes are similar, but dysbiotic microbiomes are all dysbiotic in their own way. And we've seen these sort of AKP patterns in bacterial microbiomes across a variety of systems, from humans to amphibians. And basically what happens is that when you see a decline in host health, that corresponds to an increasing variability in dispersion of the microbial community. So this indicates that this sort of dispersion is an alternate framework that we could use to try to understand what happens to microbial communities in healthy and unhealthy hosts. So there's been a lot of work in this bacterial host sector, but I'm very interested to know whether or not these AKP ideas could potentially apply to other types of host symbiont relationships, for example, a coral dinoflagellate symbiosis. So here, here's our coral again. There's been a lot of work looking at Symbiotiniaceae, focusing on trying to understand what dominant members or specific symbionts are doing within hosts. So that would be like taking this community and collapsing it down a bit so that you're focusing on the dominant types, which would be the dark blue or the yellow. And this has identified interesting specific symbionts that sometimes can have differential performance under certain types of situations, like with infection resistance or differential growth, members within these clades, or thermal tolerance. And these sorts of studies have been really important in terms of determining physiological contributions of specific symbionts to holobiont performance. So what I am suggesting is that in addition to looking at that, if we also are looking at holistic community properties, we may gain additional understanding in terms of holobiont dynamics. And so in this case, we're looking at all of the parts of the community. And in particular, this might be useful in terms of better understanding the processes that are regulating symbiont communities and understanding the sorts of communities that are associated with healthy versus unhealthy holobionts. Okay, so what we set out to try to test was whether you could see variation in these Symbiont community level properties in hosts that exhibited high stress tolerance versus low stress tolerance. And so following along with these ideas from the AKP, we were hypothesizing that under ambient conditions, high stress tolerant individuals would have lower symbiont community alpha and beta diversity, and that when you exposed communities to stress, it would increase the dispersion observed in this whole community. And so the way we tested this was using a tank-based experiment in Australia, and Dr. Rachel Wright led this work, and what she did was she took 40 genets of Acropora mallepora, or individuals, and fragmented them into 15 pieces per genet, and then she put 3 each of those pieces into all of these treatments. So addition of virulent bacteria, ocean acidification conditions, heat stress, a stress treatment with all of those combined, and a control treatment. And there was a 10-day exposure period during which we monitored the survival of all of the coral fragments, and at the end, we were able to rank these 40 genets in terms of their survival from highest to lowest. And that's essentially what you can see here. This is a survival graph where the fraction of fragments still alive on a particular day of the experiment is depicted with these lines. So each line is an individual genet. And so what you can see is, so we looked at the symbiont community properties post hoc. So at the end of this 10 days, when the experiment was over, all of the fragments had been processed and preserved. We then went back to this data set, looked at the survival rankings and said, okay, let's find the best performers and the worst performers among these 40 genets and look at the symbiont community properties within those. So we chose the best genets. Oh, oops, I'm pressing that again. We chose the best genets as those, the red lines that had very low mortality. They were high stress tolerant. And then we used these blue genets as our worst genets. These had relatively low survival. They were relatively low stress tolerant, but they weren't the least stress tolerant genets. We could not use these because they had such poor survival that there were not enough samples left to process. So then we extracted the DNA and amplified the Symbiont internal transcribed spacer 2 region of the rDNA using Illumina-MiSeq, and then processed it through a bioinformatic pipeline using Data2 and Lulu. So ITS2 has intragenomic variation, which can be problematic. Lulu is an algorithm that is fairly successful at collapsing these intragenomic variants using sequence similarity clustering and co-occurrence patterns. So it is similar to some of the methods like SymPortal that have been published recently. Okay, so I am mostly going to talk with you about the community level properties, but I will quickly show you the general dominant symbiont types that we got back. So each one of these blocks are all of the fragments for a given genet. And what I'm showing you is the relative abundance of the most dominant symbionts within each one of these fragments, which is a vertical bar for each of these treatments. And so all the bests are up top. As you can see, they're all dominated by cladocopium types, and they are relatively similar to each other. In the worst genets, you can see mortality in these black and white hatched bars. They also are generally dominated by cladocopium symbionts, and there's this one worst genet that also has some durastinium symbionts. And so all of these, except worst 27, also had other ITS2 types that were present at low abundances. And we did see, when we used some indicator species analyses, we did see some specific types popping out, like the durastinium in this worst 27 and cladocopium C3K in the best performers as being indicative of this best or worst status. And we did some additional analyses using this generalized joint attribute model that I'm happy to talk with you about later, but I do not have time right now. Okay, so in terms of the community level properties, now I'm going to show you this in terms of the worst performers, or the low stress tolerance genets in blue, or the best performers or the high stress tolerance in red. And under just the control conditions, what you can see is that the low stress tolerant genets had significantly higher alpha diversity and significantly higher beta diversity, including in terms of dispersion. And then next, we wanted to look at these best and worst performers and see whether or not when you took those fragments and you exposed them to stress, whether or not their symbiotic communities became more variable. And so with this, we had to normalize the stress treatments for a given genet because each of the genets had a different starting symbiotic community. We had to do this normalization. So we took the Bray-Curtis distance values for a stress treatment of a given genet, and we normalized them to the average control values for that genet. And so because this is a normalized calculation, what you can see here is for values close to 1, that means that there wasn't very much change in community variability in that microbial community between the stress treatment versus the control. And so for the best coral genets, we really saw that basically stress did not change the dispersion of those communities. So they stayed uniform. In the worst genets, we did see significant increases in dispersion in the bacterial addition and the heat stress treatments, and perhaps there was a trend towards that in the combined. So this is really the first indication that we had that the anacarina principle might be relevant to a coral dinoflagellate symbiosis. So just to summarize this part, we did see certain Symbiont types associated with best and worst performing hosts. We saw that each, even though each genet had different starting communities, the best genets were more similar to each other, both in control conditions and under stress. And so that makes these whole community parameters interesting to think about in terms of looking at diversity metrics as potential indicators of whether or not a particular holobiont is likely to be stable under stressful conditions or not. But this needs to be explored further in additional species, et cetera. And then the symbiotic communities in the worst genets became more dissimilar in the stress treatments. So it was really interesting to see these first results suggesting that the AKP could be important, but it'll be great to look at longer experimental durations, longer than 10 days, and also to use an experimental design that involves repeated sampling of the same individual throughout a stress treatment. And so that is actually our next step, using coral reefs in Correa, French Polynesia in the South Pacific. We're able to take this up to a much more ecologically relevant scale. And there was a bleaching event that happened this past year that we're able to capitalize on as a stress. And so this is leveraging this 3-year microbial time series that my lab has been conducting with Becky Vega-Thurber and Andrew Thurber at Oregon State University, which includes 3 coral species around the island. And so I think this will be a really nice next step for testing the AKP. And so just to show you, if one of the species that we are collecting data for is Acropora hyacinthus. And so to give you an example of these 3 different timelines we have for this timeline, we have 2 pre-stress collections, a stress time point during the bleaching event, and a post-bleaching event time point. And so for some of these, we had some tagged corals that we've been following over time that didn't bleach. There was no visual bleaching over the course of this time series. We had some that we saw bleached during the 3rd time point, but then started to recover. We had some that bleached and were still bleached by this recovery time point. And we had others that bleached and then died. And so I've given these symbols. You're not going to have time to fully process this, sorry. But basically what I'm outlining here is a set of predictions that would follow similar to the tank-based experiment, in which with these increasingly significant health declines of those different categories, we might expect to see increased dispersion of the symbiont communities out to these ones that bleached and died. And we might even expect to see at the original time points pre-stress that, that some of these that were more prone to failure or less stress tolerance may have more variable communities. Okay, so ultimately, we are excited about trying to look at these community diversity metrics to understand whether they may be predictive on a reef as a tool for understanding which holobionts are likely to perform well under stress. Okay, so for the second part, Karsten Frupstra, who is also a PhD student in the lab, has been leading a lot of this work, trying to understand how a particular viral lineage might be infecting Symbiont in corals. So first, I would like to give you a little context based on Symbiont. So we have this emerging understanding of what individual symbionts and also symbiont communities, how they might be influencing host or holobiont health or function. And a lot of the reason or the keys that behind this understanding are, one, that we have a universal set, well, we have sets of universal marker genes that are available, which means that we can go to lots of different hosts, take a sample, and identify all of the Symbiont present in that sample. And so over space and time, we can catalog the symbiont diversity in hosts, for example, in this sort of classic paper by Dan Thornhill, at this point, tracking dominant symbionts in hosts over time. And then also we have some Symbiont in culture, which is really important because we can characterize their physiological tolerances, both in the host and separate from the host. We can develop genomic resources for particular symbiont types. And then we can take these symbionts in culture and do direct experimental manipulations where we add them to aposymbiotic hosts and see how the addition of symbiont, a single symbiont or multiple kinds of symbionts influences holobiont function. So when we have a lot of challenges doing that with viruses. And one of the first challenges is just that there is a huge diversity of viruses present associated with a coral colony because there are so many partners present in a coral holobiont, and viruses can potentially infect any one of these. Different viruses could potentially infect any member of this. So when we detect viruses from a coral holobiont, it's very difficult to parse out who's infecting who and what they're doing. There are also a lot of hypothesized roles of viruses in coral colonies. And we have to be really careful here because we don't have empirical data to support any of these roles in particular for any particular virus. So they could contribute to disease signs, for example, and in fact, some T4 phages have recently been associated with black band disease in corals. Viral infections could also potentially impact colony thermal tolerance. And I think a really tantalizing analogy to make is with this panic grass fungal endophyte virus system, in which these panic grasses are able to use a very extreme temperature. There are soils that are heated to 55 degrees Celsius in Yellowstone National Park. And this panic grass can handle living in those soils only when it contains this fungal endophyte and only when that fungal endophyte is infected with this virus. So it is interesting to think about whether or not there might be some latent infections of Symbiont that could contribute to thermotolerance properties. And we have seen some viruses that are interested, that we know are infecting Symbiont and that in particular might be implicated in thermal tolerance, but this is a hypothesized role. If certain types of viruses are host specific or relatively host specific, then certain types of lytic or active infections that lyse cells could shape some of the Symbiont community patterns that we see in hosts. and maybe contribute to some of these patterns. And another possibility is that if there was an extreme event, sort of a mass lytic activity by viruses, then they are ultimately viruses of Symbiont, and they could be lysing a lot of these cells at once and potentially even contribute to something like bleaching signs. So when we think about the potential outcomes of viruses in corals, there are many possible outcomes, and not all of these are necessarily antagonistic to the host of the virus, and certainly not necessarily antagonistic to the coral holobiont overall, right? And so, because we do not yet have empirical data on demonstrating the role of a particular virus in a host or a holobiont for corals, we have to be very careful about how we design our experiments and how we interpret our data to make sure that we aren't putting a virus in a stereotype box and missing something important that it could be doing. So in terms of challenges, part of what's hard about this is we don't have a universal marker gene for any of these viruses. In part, that's because viruses can have either DNA or RNA genomes, and they also evolve very rapidly. And we don't yet have any coral-associated viruses in culture, although one of the things my lab is hoping to do is to add that resource to the field. And we haven't had any data on the prevalence or diversity of a specific kind of coral-associated virus. So there's no lineage-specific viral information available yet. What we do know is that we have seen signs of diverse viruses associated with coral colonies, but although we see these signs, the virus as a whole often is not very well characterized. And we sometimes see increased viral production in stressed hosts. Okay, so there has been a lot of work done by a variety of labs making metagenomes or metatranscriptomes and or using other tools, trying to just get a handle on the types of viruses that we typically see associated with coral colonies. And so these are some of the viral types that we believe are commonly found in coral colonies. Some of these infect or target the bacteria, coral-associated bacteria as a host. Some we think are probably targeting the coral animal tissue itself. Others we think are targeting the Symbiont. And I want to call particular attention to this HCRNAV, which we're going to come back to, or it's HCRNAV-like virus. HC stands for heterocapsid circular squama, and it's an RNA virus. So this HCRNAV-like virus is most similar to a virus of a free-living dinoflagellate. That's a positive single-stranded virus. And then there are also some viruses where we are not even really ready to make a guess yet as to which particular host they're infecting, but we see them in the coral. Okay, so our group has been very excited about taking steps moving towards getting at a lineage-specific viral analysis. So we've been drilling down into this HCRNAV-like virus, and this is essentially, in terms of new, I don't know about new term, but you can think of this as a Russian doll symbiosis or a nested symbiosis, in that this is a host with a symbiont inside of it, which I don't really have time to get into, but it's very interesting to think about like dynamics in terms of like how is this virus getting through all these layers to find its host, et cetera. But this HCRNAV-like virus looks very promising as a first host to get prevalence and diversity and distribution data on. And now I have to give a shout out to Madeline Van Appen's lab, because they used RP-CISPA to generate this set of nested degenerate primers that we can use to amplify the major capsid gene, the major capsid protein gene of this HCRNAV-like virus. Our lab has worked hard at sort of hacking into what has been a very time-consuming, low-throughput process associated with doing that, and making it so that we can kind of just like Symbiont or very similar to Symbiont collect a coral piece, put it in some preservative, and then use this nested primer set to be able to create cDNA and then amplify this major capsid protein gene. So we're very excited because now we can do sort of larger scale, look at larger scale distributions of this viral group across hosts. And so we've started just looking to see how many coral colonies we can find this virus in. And the answer is, out of what we've looked at so far, we're finding it in a lot of coral colonies. So this is from Correa, but we've also looked at things in the Virgin Islands and a few other parts of the Caribbean, much lower sample sizes, but basically we've been able to find signatures of this virus so far in every coral species that we've looked at, and in many of these coral colonies, both control and heat stressed. An interesting thing here, though, is that I'm saying there's high prevalence of this virus in coral colonies, because we're taking a sample of a coral, preserving that, and then getting these HCRNAV-like sequences out of it. So a coral fragment can have 1 to 2 million symbiotic cells per square centimeter of coral tissue. So what does it mean when you get a positive detection from that many cells? It could be that it's just a probability game, and the reason we're getting it from so many corals is that just maybe you know, at any given time, a very small number of those symbiotic cells happens to be infected with a lytic infection, and that's what we're detecting. It could also, you know, and or be that we're capable of detecting dormant or latent phases of this virus. If it does that, that could be in the cytoplasm, or perhaps it's some type of endogenous virus that's integrated into the symbiotic DNA. So it's, you know, we're very excited to be able to get our hands a little bit on one kind of virus consistently from lots of samples. But now the next step to seeing if it's an important virus in the holobiont is to really sort of drill down and start trying to amplify this from individual symbiotic cells. So that's what we're working on next. In terms of diversity and host specificity of these HCRNAV-like viruses, we did a tank-based experiment where we took 5 different coral genets, fragmented them up, and then distributed them among heat and control tanks, and then looked at colony health, the dinoflagellate community identity, and tried to characterize these viruses. And here you can see in the control fragments, the dominant symbiotic types that we saw at the end of the experiment. They're very similar to each other. And here are the HCRNAV-like OTUs that we got out of those same fragments at the end of the experiment in the controls. This is based on 97% clustering. And one thing that you can see is that despite having, you know, not very much diversity in terms of symbiotic types, we're seeing many more types of HCRNAV major capsid proteins, and these are sort of distinct among these colonies. So unfortunately, at least with this initial data set, we really can't say anything about host specificity because, you know, we don't have like variation that we can connect things up with. So can't say much there with this data set. There is a lot more to unpack and try here. One of the things we need to do to just figure out or to try to see where things might be biologically relevant with this is to try clustering these OTUs at different % similarities, which we've started trying. We can also translate them to amino acid sequences and work with those. We definitely need to look at the symbiotic communities using finer scale resolution markers. And Todd, I'd love to talk with you about that if you have time. And we also can take some of these really dominant OTUs and go and search for them in published data sets to see if we can find an indication that, oh, this particular OTU, now that we know what it is, we can see it in lots of places. So we can start looking and seeing if at any of these levels of clustering, et cetera, if we suddenly see an OTU popping out as very common. Perfect. Okay. Now what you can see is the control compared to what the symbiotic community was in the heat treatment. And as you can see, there was not very significant change over the 4.5 days of this experiment. We might not expect there to be very much change over 4.5 days, because bleaching can take longer. We had this experimental timeline set up because we were thinking about how long we thought it might be to induce some viral cycles. Maybe the most, keep your eye on this shift in colony 3, but basically all of these coral fragments appeared normal throughout this experiment. Maybe there were some bleaching process starting before visual signs were apparent. Now you can see for these colonies, here's the control versus the fragments in the heat stress treatment. And for each colony, and these arrows show increasing time points during the experiment. So what you can see is that there's a significant amount of variation. However, we did find that heat stress differentially affected the OTUs that we saw in colonies. So there's a very significant colony effect. We tried to account for this in our model by using colony as a random factor. But there also is some contribution of this heat treatment. And that's really interesting, particularly in colony 3, where maybe colony 3 is interesting. So one possibility with this is that heat stress could have triggered at least some lytic activity in some of these viruses. Perhaps they became stressed and more active. And one interesting thing is that there was a significant increase in these major capsid protein OTUs in the heat-treated colonies in terms of alpha diversity relative to the control. Okay, and so this grant is actually intended to be more focused on this type of question with these viruses. So we certainly are going to go and use this data set now to look for HCRNAV major capsid protein on a larger scale. Okay, so in terms of an overall virus picture, there are a lot of challenges associated with trying to get these viruses into our clutch and understand them, but we're making progress. Trying to culture them and being able to do these lineage-specific analyses in space and time is going to be important to understanding their roles. We really need to remember that that context is important, just like for Symbiont or bacteria or anything else. The ecological context or the environmental context that a virus is in is going to influence how it interacts with members of a coral colony. And they aren't viral infections aren't necessarily only going to cause harmful effects. So we need to limit our assumptions. And these HCRNAV-like viruses, as a more specific finding, appear highly prevalent in coral colonies, and we see some shifts under heat stress. It's going to be really interesting to look at how they might affect dinoflagellate physiology and function. Okay, and now really quickly, as the third thing, just thinking about where and how corals might get their symbionts. Hosts basically have two options for picking up microbial symbionts. The new generation can get them directly from the parent via vertical transmission, or they, via horizontal transmission, have to pick them up from environmental pools with each new generation. And about 50% of shallow stony corals are horizontal transmitters. So we've been trying to find these environmental pools for Symbiont, and it was pretty difficult, with the first papers successfully doing that coming out in about the late 2000s. And when they were found in sediments, they were found in the water column, and they were found associated with macroalgae. So those environmental pools do exist. However, interestingly, the densities of these Symbiont cells are pretty low in these environmental pools, and also the genetic types that we typically see are often different than what we see dominating stony corals. So it seems like maybe we're missing something there. And so I think vector-mediated transmission is something interesting that we can think about here as a partner that's even a potential partner that's even outside of the Matthews schematic. It's interesting to think about the fact that pollinators are really important in conducting horizontal transmission of bacterial and yeast symbionts into newly opened flowers that have sterile nectar. We don't have any pollinators down on a reef, but we do have corallivores biting coral and digesting it out. And the role of corallivores in symbiont dispersal is definitely understudied on reefs. There are 2 papers that have looked at it, and they found very interesting things, that Symbiont in corallivore feces, or things that eat corals, at least some of them are photosynthetically viable, and at least some of them are culturable. And one of the papers did get some genetic information on the types that they found, and they did find some types that were associated with corals. But in general, there is a lot more work that can be done in this area. And so my group got really excited to try looking at this in Correa, in particular capitalizing on the fact that it's a long-term ecological research station where we have lots of data on coral, macroalgae, and fish abundances over time. So I don't have a lot of time to show you this, but basically our overarching hypothesis is that corallivore feces are a major environmental pool of live Symbiont for stony corals. And so we started looking at this by collecting feces from fishes that were very different in terms of their dietary preferences. So we collected from some that are obligate corallivores, they only eat coral. We collected some from facultative corallivores that will eat coral but also eat other things. And then we collected some from fish that we know do not typically eat coral. And so we had some hypotheses about that we would see more symbiont cells in the things that eat more coral, and that we might see higher symbiont diversity in things that eat from lots of different places. And so what I'm going to show you very fast is just a stripped down data set of these species. And here you can see in the obligate corallivores, again, this is Symbiont relative abundances. And what you can see just by color is that these obligate corallivores have pretty similar symbiont community compositions compared to fossilopterid corals. When you look at this facultative corallivore, more diversity is added, starts diverging a bit more from the corals. And when you look at this no coral control, there's a lot of different Symbiont in there. I don't have time for this slide, but essentially in terms of cell densities, because it's one thing to get the DNA, but what if they're all dead? So we found a way to use a trypan blue stain to differentiate live cells in feces from dead cells, and then do counts on a hemocytometer. And the cool colors represent obligate corallivory, and going down here. And what we found is that live Symbiont cell densities are much higher in the feces of fish that eat more corals. And sediments and water would be, I don't have them on here because we don't have that data yet, but based on the literature, they'd be lower than those other species at the end. And here are just some that we cultured out of the fish species, so you can see that they're alive that way too. And so this source of live Symbiont that matches what we can see in corals is much denser than from the sediments or the water column. And then we can use all of that LTER data to try to scale up to understand how many symbiont cells might be getting moved by these corallivores on a reef. And so in this example, we calculated that about 400 million live Symbiont cells were potentially dispersed on the moraine forereef per kilometer squared per day by this fish species. So obviously this is just a start, and we're gonna have a lot more soon. But basically, in terms of a take home on this, we think that these obligate corallivore feces are potentially a very important source of Symbiont for horizontally transmitting corals. And passage through the corallivore gut and reuptake by a coral, there are more experiments that need to be done here, but this could be a common experience for a Symbiont cell. And if it is, it's really interesting to think about what a different environment it could be in that gut and in those species compared to in a coral, and all the types of interactions that it could have with different microbial symbionts or other things. And so if this ends up being important, then we need to consider these multi-partner interactions in corallivore guts and feces, as well as within coral hosts. So hopefully, I've talked with you about these symbiont community characteristics as potential indicators of host stress tolerance, where we're at trying to understand viral interactions with Symbiont, and thinking about where else these Symbiont might be interacting with other microbial symbionts. If nothing else, I hope that you have grown increasingly excited with me about these multi-partner interactions and their potential importance. And so with that, I just want to acknowledge the folks that we work with in Correa to collect this long-term data set, and also the folks in my lab and collaborators and funding sources, and all of you.
Thank you, Adrienne. That was awesome. I have a question. Go ahead.
So I had a question about when you were, looking at your capsid proteins, and you were initially doing the clustering at 97%.
Mm-hmm.
You're like, should we look at other levels? So when we think about like 16S, looking at 97%, there's sort of like biological reasons why we choose that threshold related to overall species and the capsid. Do you have any sense of, like viral research, because I have no idea about viral. A sense of, is there similar relationships between capsid sequences and overall genome sequence that say it, are they evolving quicker or slower or at the same rate as the genome as a whole? To be able to make that kind of same connection.
So essentially what I can tell you is that, in terms of this particular HCRNAV-like virus, it's only like the real HCRNAV. We need to give it some sort of name so that we can stop saying HCRNAV-like soon. So we really don't have very much information about it at all.
Do you have the whole genome sequence?
There is a whole genome sequence. But the sort of that range of 97 to 60% comes from various aspects of the literature. So the Montalvo-Proaño paper, their pipeline that they used 60%. There is from the real HCRNAV that infects that free-living dinoflagellate. There are two broad types, the UA type and the CY type, and they use this. They use the 97% clustering, and that's informative for that viral group. And then we're also, I think we've also tried 92%, because if you sort of look at, where things really start to change, depending on where you cluster, I think that's, at least in terms of seeing, what suddenly makes a huge difference in what you get out, that seems to be the 92. So that's the best I've got right now.
You may have said this. Excuse me, just in case, but did you look for them in dinoflagellate cultures or not?
So we are doing that now. So part of this has been, we have so many of these. That heat tolerant take experiment that I showed was the initial sort of test run to see if we could get this working, because I was afraid to try it with the big Correa data set as a first pass. And so it took a really long time. We had to both work on getting costs down related to the high-seq sequencing, and then also make some changes to the bioinformatic pipeline to make sure that we were really confident that what we were getting out was major capsid protein, et cetera. So the bottom line is we have 4 other projects, including direct Symbiont culture work, where we're waiting for sequences back, and so soon. Yeah. Liana?
I love the idea of what the Correa is. Especially, there's a couple of things to it. You know, you see that you've got lots of symbionts. But I had a different question. So you look at your Symbiont community plots, right?
Mm-hmm.
I don't know much about that food pipeline. It just strikes me that they're all in the same ratio. It's awesome. Like, why would you get, like, for the Acropora,
Mm-hmm.
you know, why would you get 75% of one and 10 of the other and whatever across all these comps? What keeps them in that ratio? Just, I don't know, there's just something there that just, I'm worried about that. It seems unlikely that to be able to ratio.
Well, we've tried, I mean, some of those, it gets into, one of those is like, if it's C3 versus C3K, it gets into some of the stuff of whether it's empirical. There are some details with ITS2 types that are issues that could be an issue there, but in terms of, I don't think it's the pipeline, because we've tried, we're in the process also of comparing research with and without clustering, with and without clustering in Lulu, and then this data too, with and without Lulu, and we also want to do it with SymPortal to see like the variety of these different options that are available for looking at Amplicon sequence data for the ITS2, whether or not you get similar patterns or not, and essentially across these different tools so far, there are some minor changes in terms of what gets solved, but
okay, so let's say this is just what that's what the system data told you, right? Mm-hmm. So this is what it is. But what would be a biological explanation? How, you know what I mean? It's like I mean, it just seems hard to me to figure out how a coral, presumably it would be the half host, can somehow control major ratios, different ions, exactly. I mean, it begs for an explanation. You know, even if the sequence data is exactly right, like I'm struggling how to. I mean, 50-50, I can sort of.
Well, you're talking about the first data set with like where the bests were very similar and then the worsts were more different. Right?
Exactly right. And so you said like you looked at their different genets for sure. The hosts are all different genets, so it's not.
Yeah, they did, because there's a companion paper, and they went out and used some kind of red. They confirmed that they are all distinct genets. Yeah. So I mean, I think if there had been the exact same different thing in the worsts, like the worsts were less variable than the bests. I don't know the mechanism for how the coral or the competitive result or between symbionts or what's producing that uniformity in the bests, but I mean, maybe that host is very good at modulating its symbionts.
Well, let's take that a little bit further. What if you did the host ITS sequencing using the same pipeline, same host ITS markers, and you got your results back, you analyzed them, and you got the same kind of proportions across from colony to colony to colony? How would you interpret those data? So there wasn't just one sequence; there was maybe a couple sequences, or several sequences, but they were all found in the same relative proportions from colony to colony. How would you interpret that?
I don't know. Would you be? Have you done that?
Yeah.
You share?
Yes, we've published on it. So the ribosomal array has many copies, so it's not analogous, right, to 16 mass. Very different. We're talking about eukaryotes, very different stories. You deal with ribosomal. So you just think about, so how would you then describe that pattern, that consistent patterning from individual species of having the same proportion of ITS? It's been done in insects, all kinds of things. It's not just one dominant sequence, it's a combination of dominant sequences that define the genome of members of the species.
I think, you know, so I had a table in here that shows all of the different Symbiotic types that were recovered from these different genets. And I think part of what happens, part of the challenge of I agree that there are issues with ITS, but I'm going to put a pin in that, and I'm going to come back to the fact that I think it is interesting and important to look at dominant types and try to get at specific types and how they may impact colony function, but that when we portray things that way, so like that bar chart, we look at it and we see those, we flatten everything to just those patterns, but each of those different colonies, each of those different genets has different other diversity. Some of them have Rivolium minutum, some of them have D2, some of those best have D1 and D1A, some of the worst have also those things plus Symbiotium A101. So there are other things that are present there. So I agree that there are issues with ITS too, but in terms of the symbiont community properties and the alpha and beta diversity stuff, there is additional, you know, we reanalyzed a bunch of different ways and the patterns keep falling out. So,
oh yeah, the patterns are.
I think seems like there is another group coming in, so I think we need to wrap these up and put these
If you have not, walk out the front door and sign in. There's 3 different sign-in sheets. If you're here, you should sign in. If you're not here.